Thank you very much for giving me an opportunity to present a talk to introduce where we are with the development of gene therapies currently. So currently, more than 2,000 gene therapies are in development. These fall mainly into the categories of rare diseases, as you can see here, and oncology applications. Within the rare diseases, gene therapies are being developed for ophthalmology, hematological diseases, metabolic diseases, and so on. What you can see here in this graph is that over the last few years, the number of clinical trials conducted has increased, and this is largely driven by an increase in clinical trials conducted by commercial sponsors, so by industry. And very excitingly, globally, we currently have 32 gene therapies that are approved, and this includes genetically modified cell therapies. Just wanted to quickly explain and define the concept of ex vivo and in vivo gene therapy applications. So when we talk about ex vivo applications, we talk about taking patient's cells out of the body, and these are often bone marrow cells, modifying these cells in a petri dish outside the body, and then transplanting gene cells back into the patient. And examples here are cancer gene therapies based on CAR T cells, as well as immunodeficiencies, which I'll talk a little bit more about later. When we talk about in vivo gene therapy, we're talking about injection of gene therapy agents directly into the body. And this can be an injection that's done intravenously or in the spinal fluid, can be into the muscle, or by inhalation if you want to do gene therapy for lung diseases. And examples here include diseases such as spinal muscular atrophy, hemophilia, as well as fibrosis. And again, I'm going to talk a little bit more about in vivo gene therapy applications later in this talk. Now, when we talk about gene therapy, it's important to know that we need certain gene transfer agents, or we sometimes call them gene transfer vectors as well, because we can't just sprinkle genes onto cells and hope that they enter without the help of a vector. And in this context, we're using viral vectors, so putting the gene inside a viral vector, or sometimes we're using non-viral vectors. And non-viral vectors could, for example, be a lipid, a fat particle that we're mixing with a piece of DNA that contains the gene of interest. Now, when we're talking about viral vectors, it's really important to understand that a viral vector is not a virus. And the example on this slide is viral vectors we have developed based on lentiviral vectors. And the classical lentiviral vector is an HIV-based vector. And you can imagine that a piece of families of patients would not necessarily take kindly when we say we're treating your child, for example, with an HIV virus. So what we have done is we've made these viral vectors safe by taking out a lot of elements here shown in blue that are part of the normal native virus and that lead to disease if a patient is infected. So we've taken all these out and replaced them with our therapeutic gene of interest and some regulatory elements that we need to tell the cell how much of a certain protein product to make. The big difference between viral vectors and viruses is also that most viral vectors are not replicating anymore once they are delivered to the patient, whereas the native viruses, as we all know, do replicate and cause disease. So this is a really important concept and important to remember that viral vectors are not viruses. Now, the question is, how do we choose the right viral vector? And it's certainly true that the field over the last 20 years has had a steep learning curve. And when you consider which viral vector or non-viral vector to choose, you need to think about whether you're treating an acute or chronic disease, whether you want short or long duration of gene expression, whether you are treating dividing or non-dividing cells, whether you are conducting ex vivo or in vivo gene therapy, and also the size of the gene is important because some of the viral vectors that we're using have a limited packaging capacity, which means that they can only carry comparatively small genes. So these are factors one needs to consider when developing a gene therapy translational research program. Now I want to go on and just give you some examples of successful gene therapies that have been developed. The first example I want to talk about are the CAR T cells that have been developed to treat tumors. And here you need to know that some tumors make specific proteins which are antigens, and these antigens can only be found on the tumor cell. What our immune system does is target these antigens in an attempt to kill the tumor cell. Our immune system tries, but is not often very good at this. So what we can do using gene therapy is make the immune system, and particularly cells called T cells, better at recognizing and killing the tumor by arming them with a specific protein that recognizes the tumor antigen. And these armory, if you like, are called chimeric antigen receptors, which have fairly complicated structure that I'm not going to go into today. But what's important is this red area here on the chimeric antigen receptor. This is the area that will recognize the antigens on the tumor cell and bind to these tumor cells and then lead to killing of the tumor cells. So the way this therapy works is that cells, in this case T cells, are taken out of the body of patients, which is a very simple process. They are then modified in a petri dish by using a viral vector that carries this CAR receptor. And then you have these genetic modified cells here that are now all expressing proteins on the cell surface that will recognize the tumor cells. These cells are then multiplied again in a petri dish, retransplanted into the patients, where they will find the tumor cell, recognize the tumor cells, and kill the tumor cells. Two examples of licensed CAR T-cell products are listed here. One was called Kymriah, the other one is called Yescarta, and they were licensed in 2017 or 2018. And these products are developed specifically for B-cell lymphomas, and they've certainly shown that treatment of patients with these products leads to very significant improvement in survival rates. They do have side effects, but now that we know about these side effects, clinicians are able to treat the side effects very efficiently. What we don't at the moment, and only time will tell, if these gene therapy-based drugs will significantly reduce relapse rates over time. And this is something that will be followed and data will be collected on. I think future challenges in the field of CAR T cells in the context of oncology are related to the transition of autologous cells to allogenic cells. And by this I mean autologous cells are cells that are taken out of the patient, genetically modified, and then transplanted into the same patients. So the medicines are developed or manufactured, I should say, on a patient-by-patient basis, which is never a good thing from a cost and time perspective. What would be much better is if we could develop universal donor cells that could be delivered to all patients, could be ready in the freezer to use. And that's certainly a sort of challenge in the field at the moment. The other challenge is the development of CAR T-cell therapies for solid tumors. And there's an awful lot of research and many groups are working on this to make CAR T-cells efficient for treatment of solid tumors as well. The next disease I briefly want to talk about is a disease called spinal muscular atrophy, or SMA. This is a disease where babies are affected, and it's sometimes called floppy baby syndrome, because these children have an inherited neurological disorder which leads to rapid loss of motor neuron function, and more than 95% of these babies die before they reach 6 months of age. The treatment that was developed here is called Zolgensma, which was approved in 2019. And this is a viral vector which carries the SMA gene and is injected intravenously. It has led to a very significant improvement in motor function in these children. It's not a cure, but it's a very significant improvement in motor function, which now leads to longer survival of these children after treatment. One issue that I want to highlight here for the first time is that gene therapy drugs are very expensive. For example, Zolgensma here has a list price of around 1.8 million pounds per dose. And this is something I will come back to a little bit later again. Another example of gene therapies that are being developed is for blindness, and here a disease called Leber's congenital amaurosis has been very successfully targeted through a drug which is called Luxturna. Again, Luxturna is a viral vector carrying the gene that's mutant defect in Leber's congenital amaurosis. This vector is injected into the eye of patients and leads to a significant improvement in vision. Another example are gene therapies that have been developed for hemophilia. Again, a viral vector carrying either the factor 8 or factor 9 genes, which are defective in patients with hemophilia, are injected intravenously. And this treatment has led to a reduction in bleeds, as well as to a reduction in protein replacement therapies, which is the standard of care that hemophilia patients are treated with. The two gene therapies that have been developed here are called Hemgenix for hemophilia B and Roctavian for hemophilia A. Now, when Roctavian was initially licensed, it received a lot of interest in the press, and this is an extract from Scientific American, which states that the treatment for hemophilia, which was just licensed, is the world's most expensive drug and has a list price of about $3.5 million. When looking at this incredibly high price, it's really important to consider what the normal standard of care for a patient with hemophilia will cost. And the company that developed the drug very quickly stated that the costs are justified because the gene therapy drug would save the US healthcare system approximately $5 to $5.8 million per person treated. And these costs here are based on the fact that patients that suffer from hemophilia, the average treatment cost if these patients receive hemophilia B protein are roughly $700,000 to $800,000 per year. So the very high cost of the gene therapy-based Hemgenix drug will pay for itself in a very short time. My final example that I want to talk about is a gene therapy that was developed for an immunodeficiency, and this immunodeficiency is called ADA-SCID. Babies born with ADA-SCID do not have an immune system due to a genetic defect and have to grow up in a very sterile environment. Otherwise, they would very quickly die from infections with which they cannot fight due to the lack of an immune system. It's an ultra-rare disease with 1 in 500,000 babies born affected. And again, the gene therapy that was developed here is based on taking bone marrow cells out of the child, treating these bone marrow cells in a petri dish with a gene transfer vector. These cells are then retransplanted into the patient and will cure the patient for life. And This is again a treatment that has been very successful and was licensed in 2017 under the name of Strimvelis. Again, a fairly expensive treatment, but it's anticipated that babies only need to be treated once in their life to completely cure the disease. Now, Strimvelis, just in illustrating the Strimvelis story here, which is classified as a market failure, and the reason for that is illustrated on the slide. Strimvelis was initially developed by a charity, the San Raffaele Telethon in Italy, and then bought by a big pharma company, GlaxoSmithKline, in 2010. Strimvelis received market approval in the EU in 2017, but the same year GSK announced that they would sell Strimvelis. Strimvelis was then by another pharma company, Orchard Therapeutics, in 2018, and only 4 years after buying the drug, Orchard announces that they will discontinue investment into Strimvelis because it's not profitable to produce this drug, because the ADA SCID disease is such a rare or ultra rare disorder. So here we are now left with a disease that can be very, very successfully treated with a gene therapy drug, but because of the high costs of the drug, it's not a commercially viable investment for these pharma companies. And this was a big disaster for affected children and their families, as you can imagine. And what happened then is that a charity has taken on responsibility for making Strimvelis available to affected patients in Italy. So this is an example of a drug that is not successfully licensed by Big Pharma because the disease is so rare. And that's a general problem in the context of gene therapy that I'll talk a little bit about more later here on my conclusion slide. So in conclusion, future is bright. I think we have shown that gene therapy can work. We have a number of licensed drugs that patients can get treated with. So I think it's a really exciting field which has a lot of potential for treating incurable diseases. However, there are also challenges ahead, and manufacturing of these gene therapy drugs is a big challenge, and there needs to be a lot of research and development to improve manufacturing to reduce the cost. Long-term follow-up for some of these gene therapies is also a challenge, and where the healthcare system has to work out methods to make long-term follow-up feasible and reduce the burden of long-term follow-up for patients. I think a big challenge in the field will be the transition from rare to common diseases. If we have gene therapies for common diseases, it will certainly be more attractive for industry. But the question then becomes, how can healthcare systems afford fairly expensive treatment for common diseases such as diabetes? And this is where I think we need to start thinking about alternative reimbursement models to make sure healthcare systems are not overburdened by the cost of gene therapy. Which we also need to address the problem of market failure for diseases that are not necessarily making a lot of profits for industry. We need infrastructure, we need hospitals that can deliver the gene therapies. And this also is linked to the fact that we need workforce, and particularly NHS workforce, that is sufficiently trained to deliver gene therapy-based medicines. And we need very significant patient and public involvement and education in this space. With this, I will conclude, and thank you very much for your attention.